Okay, so we're starting to record. Okay. All right. And then after, we'll spend about half the time on development and evolution, which is the last topic of, I think, a very exciting course. But at the end of it, I'd also like to spend some time doing some review for the last midterm. Of course, Sean has her official review session on Sunday as well. Okay, so, but this is the last topic that will be covered in the exam, and last topic at all. And I really chose this as our last topic because it's really about a more long-term view of where this field is and where it was and where it's going, right? So, I mean, the cognitive neuroscience in general is the study of how the brain enables these functions of the mind. And we know that the functions of the mind and what that requires, to cope with a busy world, has really changed over time, right? And so the brain changes, and a big part of it is just we grow up and then we grow old, and the brain changes as part of that. So these are just maturational constraints, developmental constraints. But then another part of that is that your demands are changing, right? So while you're when you're living in the modern world, right, what you need to do to survive might be different from 300 years ago, what you needed to survive. And so our brains are constantly adapting. And so when we're talking about brain development and evolution, we're talking about these lasting changes, right, that are not just momentary. It's not just like you learned something and suddenly your brain changed a little bit. We're talking about longer-term differences. And so the major buzzword, right, is neuroplasticity, or brain plasticity. And that refers to changes in the neural pathways and the synapses as a result of changes in behavior as well as environment, and also the cognitive processes that might be going on. On the other hand, these neuroplastic changes can also result from adaptations due to bodily injury, right? So a classic example is from the motor system, when we're talking about somatosensory and motor system. Veterans who've had their arm amputated, for instance, their brains rewire, right, to accommodate differences in their body structure, right? And so those kinds of changes are definitely part of what neuroplasticity means, but it's not really a normal kind of trajectory as much as it's a change that results from bodily or brain injury. So another great example of neuroplasticity at work might be when you have a brain injury, right? Not an amputation, not a body injury, but a lesion in your brain, maybe because you fell off the bike or some kind of brain trauma. This can sometimes result in other areas of the brain kind of taking over the areas of the brain that had impaired function. So when we're talking about neuroplasticity, there's two major classes of neuroplasticity. The first is structural neuroplasticity, and the second is functional neuroplasticity. Right? And structural neuroplasticity is generally harder to come by, and because that refers to actual changes in the gray matter and the white matter of the brain. Right? So gray matter could mean that the cells, neurons, actually cell bodies of the neurons, became denser or became larger, or there became more of them, right, actual neurogenesis. But white matter changes could come from cells becoming more myelinated or becoming more coherent as time goes on. Right? So these are actual anatomical changes that go under the bracket of structural neuroplasticity. And then in contrast, something that might be a little bit shorter term and more flexible is functional neuroplasticity. And you can think of that as regional activity changes, right? So areas of the brain becoming more or less active over time as a result of learning, right? Or you could also think of them as synaptic activity. And so the classic saying, cells that fire together wire together. So that's a really good example of synaptic plasticity, where different neurons become more or less connected due to the synapses that connect them. And when we're thinking about brain changes, there's a number of critical events that happen throughout brain development that really change the morphology, or the structure of the brain. So this is quite a general overview all the events that go on, well, not all, a lot of the events that go on throughout our lives. Right? So from the moment of conception through being born and through your late in your life, there's actually been more and more studies that have looked at normal brain development. And so there's major longitudinal studies funded by NIH and IMH and so on that trace the changes in the brain from toddlerhood all the way through old age. But then there's also studies that look at prenatal MRIs, and these are hard to come by, right? Because the effects of MRI on infants and fetus are unknown. So a lot of times people are only able to access the fetus in the MRI when the mother has a health problem or something that requires that they get an MRI. But nevertheless, you can see a lot of events are actually going on from the moment of conception. Right? So first of all, you can start with the forming of the neural tube, right? And so that happens at just 2 weeks, right? And so that's really just cell division, right? So kind of general biological changes. And then something that becomes more and more kind of specialized about the brain is neurogenesis. So that's actually growing of new neurons. And that happens from around week 8 through about week 16. But it actually goes on. So I mean, these little lines are just like kind of guidelines for when these things happen. So don't think of it as like an open and shut kind of window. But they're generally when most of this happens. And so a lot of brain growth happening in utero. It's really funny to think about how all this stuff is happening in your brain before you even got born, right? And then a lot of synaptogenesis, right? So that means the generation of new synapses, not just new cells, not just new neurons, but the connections between neurons. Right? But a lot of this you might not be so obvious, but it's about brain migration actually happens. And when I mean brain migration or neuronal migration, I don't actually mean that neurons are moving around inside your head. I just mean that the synapses, the axons, axonal projections of individual neurons are kind of finding their right place, right? So that means that a lot of white matter connections are involved. And so areas that are kind of distally connected are not in the same area of the brain, but connecting different parts of the brain are starting to fall in place. This is also when you can start to see major brain structures, even in the ultrasound. And then a lot of what also happens, which is kind of counterintuitive, is programmed cell death. So those of you who've taken biology probably heard about apoptosis, right? So that's just cell death that's happening early on. So it's not just that we're just growing more and more brains as we get older. There's actually a lot of pruning. Pruning just refers to the programmed cell death as well as neuronal migration as well as myelination. The development of synapses. Okay, so that's all of the things that happen even before you're born. I think that's really quite amazing. But after you're born, what happens, right? Okay, so by the time you're 2 and onwards, you can officially participate in MRI studies. But it's very hard to imagine one person coming back for the same MRI scans every year. But what people have done is take these longitudinal views of the brain, right, in order to try to identify a major trajectory of brain development over many, many years. Okay, so on the top left, what I'm showing you is a view of how these results kind of work, right? How the researchers were able to plot these developmental trajectories. Right, so each subject is 1 point, right? And if they came in multiple times throughout several years, they would show up as connected 2 points, right? And so you can see that there's one subject right here who came in around age 4.5, right? Oops, and then they came back again around age 6 or 7. And then there's another subject who came in for the first time at age 14, and then again at age 20, and again at age 21, right? So you can see how when you have lots and lots of these data coming in, well, first of all, you start to see these trends, right? So female brains tend to be smaller than male brains in the cerebrum, and that's just because female heads are smaller than male heads. But it actually depends a little bit on where exactly in the brain we're talking about, and I'll talk a little bit more about that in a bit. Okay, so and then the other nice thing that you can see is that once you have enough of these data points, you can start to project a smooth curve, right? So instead of total cerebral volume, you can also do the same for white matter. Right? So these are just curves that are smoothed out versions of all of these different data points over time, right? And so by having a few people coming in several years apart, and then also lots of people just coming once every, just for a one shot, you can start to make these major clear trajectories. And what you can see is that white matter keeps developing. This is the other major finding from this big study, is that white matter keeps developing even late into adolescence. So the white matter volume is increasing overall, whereas gray matter actually kind of tops off at around age 10 and a little bit later. Right? So by the time we're about age 10, we basically have all the gray matter volume that we need, but the white matter volume is still developing. So I think that's another major finding from this work. And then the other really interesting difference is that white matter changes over time, it increases over time. Gray matter kind of tops off over time, but the frontal lobe actually tends to come online later and be even later in myelination compared to other parts of the brain. And so just another snapshot of white matter development. Here I'm showing you sagittal views of the corpus callosum that show the corpus callosum from very early on in life all the way until adulthood, right? And so you can see that when the baby is full term, right, so 37 weeks, you can already see that there's the gray matter, white matter areas like the corpus callosum showing up, but as they grow older, you can see that the whiteness, right, of the white matter, especially just focus on the corpus callosum, you can see how that distinction between the whiteness of the white matter and the grayness of the gray matter becomes a lot more clear, right? And so by the time you're an adult, where you can really see these sulci and gyri that are quite clearly differentiated. And I think it's a really nice kind of snapshot of how the white matter is relatively late developing in life. And then another way you could visualize these data, right, is you can compare the different areas in how much they've changed over time, and you can make a colorized plot so that the colors correspond to the relative time of development. And so this is exactly what this is. That's the color in these brain images are showing the amount of development at each age. Right? So the amount of changes in gray matter volume. And what's really striking here is that the prefrontal cortex, right, actually doesn't really show all these changes until relatively late in development. And so that's another way of saying the same thing. If you're looking at neuronal migration, and then also at synaptogenesis, and also at cell death, and then eventually at brain myelination, you can see that the different areas of the brain can develop at different rates. So the areas like the somatosensory sensory motor cortices, so everything that's like primary areas of the brain, primary visual areas, primary auditory areas, primary sensory and primary motor areas, they are fully developed relatively early on, right? Whereas the parietal lobe and the association cortices are kind of in between, and the prefrontal cortex really is the latest in becoming fully mature. And so I think that kind of dovetails quite well with some of the work we're talking about when we're talking about the cingulate cortex, right, which is part of your executive control system. We're talking about how children with ADHD and also teenagers have some trouble inhibiting their responses, so have some trouble with executive control, and that's related to this relatively late myelination and development in the prefrontal cortex. Okay. So what we're seeing across many different studies is that there are decreases in cortical gray matter, right? That you actually start with quite a lot of brain matter by the time you're born, but between childhood and adulthood, you're actually seeing overall decrease in cortical gray matter, but still general increase in white matter as you get older. And in fact, there are even later in development, it's not until very late in development, like your late teenage years, when your prefrontal cortex gets fully developed. And what I would like to point out is that, I mean, these things are not just, there's not a one-to-one correspondence between when you start, you know, the way your brain looks when it starts, and the way it becomes as a fully grown adult. And of course, that's because we all use our brain differently. And so there are these brain changes that are due to use. And so that's called use-dependent plasticity. And that can refer to structural neuroplasticity or functional neuroplasticity. Use-dependent plasticity just means changes due to use. And so a really nice example, I think, comes from looking at the arcuate fasciculus, which is a major white matter pathway in the auditory motor system. And so, of course, if you think about who uses the auditory motor system a lot, there are people who sing, people who play musical instruments, right? They have to listen to sounds and they have to react to sounds. And so what this study was, it's a cross-sectional study that compared the arcuate fasciculus in singers, right, people who have voice training, and instrumentalists, right, people who have like violinists and pianists, and people who have musical training but not necessarily vocal training. And then non-musicians, right? So people who don't have so much like formal musical training outside of normal schooling. And the general findings were that singers, so this is, I'm plotting the volume of the arcuate fasciculus, and we had segregated or segmented the arcuate fasciculus into a few different branches, right? So there's superior and inferior, dorsal and ventral branches in the left hemisphere, and the same in the right hemisphere. But the general findings were that while singers tended to have larger volume in the left arcuate fasciculus, both superior and inferior, but both groups of musicians, right, regardless of what kind of musical training they had, showed a larger right hemisphere arcuate fasciculus. So it seems like the right hemisphere arcuate fasciculus might be more important for any kind of pitch-related processing. The left hemisphere arcuate fasciculus might be a little bit more important for things that involve oral motor connectivity, or even the diction that comes in vocal training. And so this is just a one-shot cross-sectional study where we recruited people with different groups of different types of training and came in. You could also say, well, maybe there's a nature-nurture problem there. Like maybe people who started out with bigger arcuate fasciculus then self-selected to receive musical training. But another group has actually now replicated this in a longitudinal study where they brought people into their lab and they gave them essentially Guitar Hero. You know what Guitar Hero is? Yeah. So it's like a musical training video game. And after a week of Guitar Hero playing, people actually showed a larger right hemisphere arcuate fasciculus. So I think that's really like compelling evidence for use-dependent neuroplasticity, right? In other words, that there are actually some brain changes as a result of what you get trained on. All right, so I want to talk a little bit more about brain evolution, not just over a lifetime, but across different species. And in general, we'll see that, this is not surprising, larger animals have larger brains, right? They have larger heads, larger brains. So body weight in general is correlated with brain weight. However, there are some people who, and in general, the higher vertebrates tend to have a larger brain weight relative to their body weight than the lower vertebrates. But there are some animals, like humans and porpoises, that have an abnormally large brain weight relative to what's predicted by their body weight. So it seems like we think humans are quite more intelligent. Porpoises are supposedly quite intelligent. But so does brain size actually mean intelligence, right? But it turns out that brain size does not absolutely scale with intelligence, right? Because if you compare cows and macaques, right? Cows they're less able to do lots of cognitive tests compared to macaques, but macaques have smaller brains than cows. Then there's also abnormal differences in brain size and brain volume in human literature that can be a little bit informative about this potential relationship between brain volume and intellectual performance. So really interesting cases of microcephaly, which is just a abnormally small head size. And this is actually an outbreak that happened just a couple of years ago when the Zika virus broke out in South America and also in parts of North America, was associated with microcephaly, right? So these women who were pregnant and contracted the Zika virus while they were pregnant, then later were much more likely to give birth to babies who have abnormally small heads, right? And so this is it was a world epidemic that's since then been under control, thankfully. But I think it's quite interesting to see what a kid with microcephaly, how exactly their brains are different. And so compared with a normal head size, a child with microcephaly has a very shrunken head. They also have abnormal seizures and lots of developmental delays. Right? And this is also correlated with a microcephalin gene. So before the Zika virus outbreak, people thought this was purely genetic, but it turns out that there might be some epigenetic influences as well. And so this is a brain scan. I think it's really striking. A brain scan of a 13-year-old with microcephaly compared to an 11-year-old who's typical developing. And so, it's obviously a really striking difference. And you can see how it's all over the brain is generally decreased in volume, but I think it's different in different children. But in general, there's a major reduction in volume. And so you might conclude from there that a larger, if you have a smaller brain, you have less intellectual capacity, because these kids with microcephaly have impaired intellectual abilities. But children with autism actually have larger brains, so it's not necessarily the case that bigger is better, right? And so another possibility is that it's not just about brain size or brain volume overall, but it's about volumes of specific areas, right? So specifically, if you look at humans compared to all our cousins and other animals, humans tend to have not just the largest brain, but also larger frontal lobes relative to all the other major areas of the brain. And so this is just comparing the ratio of frontal lobes to the rest of the brain. And so from this, you might conclude that maybe it's about the ratio of frontal lobe that gives rise to intelligence. But it turns out that the story is not that simple, because it turns out that it's not just about increase in frontal lobe volume or relative frontal lobe volume, but it's also about increases in structural connectivity to your frontal lobes. So this is specifically comparing humans and chimpanzees, and you can see how if you're looking at just the same slice, right, that's coronal slice across the prefrontal cortex, comparing a chimpanzee and a human, it's not just that cortical area is different. They're actually about the same between humans and chimpanzees, but the humans actually have more white matter. Right? So it seems like white matter connectivity, right, to the frontal lobes is something that's even more accurately tracking these differences in cognitive ability. And perhaps, then this has led a lot of people to ask, right, what is it that's a sign of intelligence, right? What does it mean to be intelligent, and what is it that's unique to humans? And so, a lot of people have thought that, because human beings are the only species that have language, at least have the kind of syntactic and semantic structures that we use on a daily basis, and that we learn effortlessly across the whole population. Perhaps the ability to have language is actually what's special about humans, and that's and people have then looked at areas of the brain that are important for language processing and production, and tried to see whether other animals have to what extent other animals have the same kinds of brain apparatus. And just general finding, right? So I'm just reviewing this a little bit, right? So since we have tests coming up, right, this is useful review. Remember, this is Norman Geschwind, who posited the 20th century. I think of it as a 20th century model of language in the brain, right? So he thinks that there's Broca's area from good old Broca and Wernicke's area from good old Wernicke, and then there's the archaeophysiculus that connects the Broca's and Wernicke's area, right? And so people have gone on to ask, and we know that if you don't have an archaeophysiculus, you can't repeat words, sentences that are spoken to you. So archaeophysiculus turns out to be quite an important seat for integrating different areas that are important for language. And so people have then gone and asked what, to what extent is the archaeophysiculus conserved across different populations, right? So that means, do humans have the same archaeophysiculus as monkeys, as rats, as chimpanzees, right? So, and so this is a really nice summary, I think, comparing human brains, chimpanzee brains, and macaque monkey brains. And so the green stuff is the identified archaeophysiculus, and you can see how they're shown to be connecting. The numbers here are just Brodmann areas, right? So we know that the inferior frontal gyrus, which is Broca's area, includes Brodmann area 44 and 45. They're together the Broca's area, and then Wernicke's area includes generally area 22 and 37. And so what they're seeing is that while humans are actually the only ones who have this nice longitudinal portion of the archaeophysiculus that then bifurcates into the superior temporal portion, right? So it goes into the superior temporal gyrus and then into the middle temporal gyrus, right? So in humans, we have this nice, nicely connected archaeophysiculus that has longitudinal portion right, throughout. That's the association fiber that goes all the way along the longitudinally along the brain, and then in the temporal lobe it splits into STG, superior temporal gyrus, and then MTG, middle temporal gyrus. Right. So in contrast, the other animals actually don't have the middle temporal gyrus portion. Okay. So chimpanzees actually have connections that go into the superior temporal gyrus, but not really into the middle temporal gyrus, right? While we humans have all the way along the middle temporal gyrus, chimpanzees don't have it, and the macaque monkeys don't have it at all. So I think that's really quite interesting that macaques just lack the middle temporal gyrus portion of the archaeophysiculus, and because this area of the brain is very important for language processing, especially for semantic processing, whereas the dorsal pathway, that's the longitudinal portion, is very important for articulatory, vocal motor functions. We think that there might be some differences in auditory motor connectivity in non-human brains that lead to this lack of ability to do vocal motor entrainment. And by vocal motor entrainment, I just mean learn by imitating, right, other animals or conspecific vocal patterns, right. So when you're when I'm a baby trying to learn from you guys, right, I might mimic your vocal motor patterns, and so that becomes babbling, and eventually the babbling becomes mature speech. And so that is all part of vocal motor entrainment. And so this idea behind this paper might be, is that maybe the lack of this vocal motor apparatus means that they're not able to learn and learn speech by imitation. And so some further evidence for the importance of this archaeofasciculus in brain development and evolution quite generally comes from people with developmental delay. And so these are children with general developmental delay, and they also, turns out, they don't have as much of this middle temporal gyrus portion of the archaeofasciculus. Right, so this is a diffusion tensor imaging study looking at images of white matter in the archaeofasciculus of typically developing children. You can see that the archaeofasciculus is quite well defined there. In contrast to children with developmental delay, they still have the longitudinal portion, but it just doesn't go into the middle temporal gyrus. Okay. Yeah, so I think that's just a kind of overview of development and plasticity and evolution in the brain. If you're really interested in this topic, I know that there's Professor Treloar teaches a developmental neurological disease course, and so we will talk more about different kinds of developmental neurological disorders. And that includes things like autism and dyslexia, ADHD, right? A lot of different disorders that you might find interesting. So I think that's actually all of the stuff that will be officially on the test. I just want to do a little bit of an overview or kind of tying up of all the loose ends. When we started this class on YouTube, I started by talking about how modern cognitive neuroscience is an increasingly interdisciplinary field, right? So I know that some of you are psych majors, some of you are neuroscience majors. We don't really have a cognitive science major, but we do have a cognitive science concentration if you take a certain cluster of courses within psychology. And we don't really have a neurology department because most schools don't, but it's more considered a branch of medicine, right? So if you end up going to med school, I don't know how you're pre-med. If you end up going to med school, you might end up doing a rotation in neurology where you'd actually get to see, for instance, a person with Broca's aphasia. And that's when you're like, wow, the textbook just came alive. I mean, I think it's really kind of dramatic when you actually interact with some people who might behave differently as a result of brain injury. And I think that the idea that things are interdisciplinary really isn't going to go away. And so there's a lot of interest right now in translational medicine and things like searching for biomarkers that might be predictive of disease functions, right? So it used to be that the DSM, right? It used to be that you had a list of symptoms, and if you had 4 out of 9 of these, or 5 out of 9 of these symptoms, then you kind of qualify as having a certain disease. But now we're moving more towards a certain kind of search for biomarkers. So a nice, a really, I think, useful screening tool for instance, for Alzheimer's disease, might be, do you have plaques and tangles in your brain? If so, you either already have Alzheimer's or you're going to develop Alzheimer's. And so that helps us increase our predictions for what might happen to a patient over time. And it also helps us better identify possibilities for recovery and for encouraging brain plasticity to kick in. Right? So imagine if you have a couple of different patients with Broca's aphasia, and one of them has a larger right hemisphere archaeofasciculus, whereas the other one has a tiny right hemisphere archaeofasciculus. Well, you might expect that the person with more of a right hemisphere archaeofasciculus might be better recovered over time because their right hemisphere is better able to take over the left hemisphere archaeofasciculus that's not there. And so, kind of increasing our imaging techniques and what will be useful in a clinical setting, as well as just for research settings. Then there's also the whole industry of neurocognitive training. And people are always trying to identify treatments that are neuroprotective for that delay neurodegeneration, right? So So the work from Alzheimer's and other kinds of neurodegenerative disorders like frontal lobe dementia, frontotemporal dementia, it's pretty clear that these things aren't going to go away. It's not just going to reverse itself. But if you can find some treatments or some interventions that are neuroprotective, you might delay that progression by a bit. And then another kind of line of neurocognitive training looking at younger children, trying to ask how you can stimulate neuroplasticity. And I think that it doesn't necessarily have to be as neuro-heavy as people are making it sound, but certainly from what we know from cognitive science, right, there's a lot that you can do to help people string certain concepts together that might kind of encourage engagement and interaction, and anything that does encourage engagement and interaction is going to reward the dopaminergic system, and that's going to make children want to play, want to keep doing this more. And so the brain training fad, I think, is still fatty, has not yet gone away. But of course, I think we all have to be kind of responsible in what we claim that we can do. And so I think the field is generally learning over time. Then I think some of the work that I talked about very early on in this course, when we're talking about brain imaging techniques and brain stimulation techniques, actually translates quite well into the field of brain-machine interface. So BMI means brain-machine interface. And so when we talked about multi-voxel pattern analysis, like the Searchlight models of looking at functional MRI data, there were sophisticated statistical methods, machine learning and so on. You've probably heard buzzwords about that, but they are really useful for kind of simulating or building a model of your brain and then simulating some new outputs, right? And so being able to find new ways to decode from the brain something that can be useful in predicting behavior. And so by the machine learning work and also network analysis that considers groups of areas of the brain together can be useful for designing things like neuroprosthetics, right? So arms for people who can't use their normal arm, just by reading from the activity of different motor areas of the brain. So I think that's all I have to say, except I'm going to now switch a bit to, switch over to, I'll keep this recording going on. I will switch to my review. Does anyone have questions while I do this? Okay, so we all know we have an exam coming up, so I do want to make sure that I give a little bit of review and just like outline the topics that are important that are going to be covered by the exam. Right? So anything after the day on memory is fair game. So that starts with the work on learning. Everything on language is covered. Decision making, executive function, social cognition, right? And then the development and evolution, which we covered today. I had a slide on memory here, but it's actually not going to be covered. So I'll just remove that before I post this online. But learning, so we talked about the different theories, like things like statistical learning, serial reaction time findings, the brain areas that are important for learning. So that includes the basal ganglia, and then also the perisylvian regions. And a lot of the paradigms come from sequence learning, right? So things like artificial grammar, and also from language learning. So you need to know about the different kinds of predictions with different kinds of probabilities that are kind of built into these statistical learning types of tests. And then we spent a bit of time talking about learning disorders as well, right? So dyslexia is a pretty major one, right? So what are the kinds of dyslexia, the different theories behind dyslexia, what are the different findings, and then also dyscalculia, right? What does it mean, right? What are the general kind of lab studies that can show really clear results from people who have trouble with calculations. Okay, language is a really big topic. The neuroanatomy, I mean, I think I talked about a couple of different models of neuroanatomy. Obviously, the 20th century model that I just went over again and then also the 21st century model, right? So how does the 21st century model differ from the 20th century model? And what are the additional regions that are involved, right? And then the other kind of major topics are just actual topics within linguistics. So things like phonology, right, which is the sounds of the language. How do we learn the sounds of the language? Right? Syntax, how we put words together, right? What word order follows what, right? And how does that depend on different languages? Semantics, right, the study of meaning. And so what are the major neural findings, neural signature of semantics and violation of semantic knowledge? And then pragmatics, which are things like jokes and turn-taking and disfluencies, right? So the practical uses of how language happens in daily life. And then also, I talked a little bit about bilingualism. I think of it as a really interesting way of looking at individual differences, right? And I think it's also quite striking how different languages can vary in the sounds that they use, right? And then depending on when you learn certain languages, you can have these very dramatic differences in the brain. Okay, then we spent a good amount of time talking about emotions, right? So the general theories, right? Are emotions discrete? Right? How many basic emotions are there? Or are they continuous, right? Is it just like different points in the space? And in which case, what are the different axes of the space? How many axes are there? Right? So these are theoretical questions, and different models, different ways of thinking about emotion. Then we talked a lot about psychophysiological methods, right? So things like heart rate, skin conductance. You need to know what they do and why they're useful. Like what are they telling us about the body that might be useful for telling us about the brain. And then the anatomical structures, obviously, the limbic system is very important, and then orbital frontal cortex. And we talked, I think I showed that video of Damasio talking about his orbital frontal cortex patients who couldn't decide. And I think that's quite profound, I think. Then I also did a little bit of kind of an overview of positive neuroscience. I think that we shouldn't forget that part as well. And I think of it as a kind of extension of the work on emotions, but taking a more or less depressing bend to things. And of course that's a nice way of kind of relating the theoretical work to the psychophysiological data as well. Okay. Then we talked about decision making, which I really introduced that as a way to evolve or to think about engaging the motivation and reward circuitry, right? So the dopaminergic system, what are the areas of the dopaminergic system, and how do they connect to each other? And then there's, in contrast to the more neuroanatomical models, there's also the more mathematical models. So what's a rational choice model? What are rational choices, and when do we deviate from the rational choice? And then I talked about the reward prediction error as a kind of way to tie together these different, like the mathematical and the neuroanatomical, right? Because it really specifies how the dopaminergic system codes for choice. Right? And then the effects of uncertainty and how people respond to uncertain or imagined decisions that we might make. And then finally, social decision making, right? Things like different morality problems, right? Two-person interaction and those kinds of problems. So I think that, I could go on and on, but I'm just going to stop there, just with regard to like how I generally cover the whole topic, because all you need to do is like go back to my slides and look for the outlines, and like these are just the same as the outlines. And so you should. I don't want to like kind of keep reciting things I've already recited before. So I'm going to give you a couple of practice questions, and then I'm going to go over like a few general like administrative things. Yeah.
I just got a quick question from the emotions
Mm-hmm.
part that you're talking about. So within the orbital frontal cortex,
Mm-hmm.
you talked about dorsolateral and ventrolateral areas, and I just wanted to know the difference.
Yeah, so generally, dorsolateral prefrontal cortex are more responsible for rule enforcing, right? And for memory-based areas. So those are, when people are talking about like, oh, working memory-based areas, they're talking about dorsal, lateral, prefrontal cortex. But I think that the real tension is between, the really interesting story is between the dorsal and lateral prefrontal cortex and the orbital or ventral medial prefrontal cortex. Yeah, so the ventral lateral prefrontal cortex, I mean, that includes like the Broca's area, so that's like kind of its own other territory. But this lateral versus mesial question, like kind of contrast that kind of keeps getting highlighted in the work on emotional decision making. That's more about the dorsal lateral prefrontal cortex. Yeah, that's a really good question. Yeah. Okay. Any other questions? Okay, so here is one. The dual stream model of language posits that dorsal and ventral pathways are responsible for what and what processing respectively. So is it for articulatory and lexical? Is it for lexical and phonological? So who thinks it's A, articulatory and lexical? Okay, great. So yeah, so most people right. So yeah, so I think the way to I think about it is that the dorsal system goes towards the motor cortex, which is important for articulation, because motor, you use your motor parts of the brain to articulate. Whereas the ventral areas are it goes towards the anterior temporal lobe, right, and that's very important for finding words, which is the lexical semantic processing. So what would not be right is, so articulatory and sensory motor are kind of the same thing, right? Phonological is almost only in the early parts of the whole system, right? So almost only in the superior temporal lobe, whereas conceptual processing is, you could think of it as part of the ventral system, but so that's not really a real dichotomy. And then there's lexical and phonological. And again, phonological is so much more kind of basic, right? That's more acoustic. So I wouldn't really think of that as a part of that dichotomy. Then there's the spectral and temporal is really, I really think of it as spectral processing has been associated more with the right hemisphere auditory areas, and temporal processing is more left hemisphere. Right, so that's not so much dorsal ventral. Okay, here's another example of a short answer question. So which areas of the brain develop early in life? Which develop late? What techniques do the studies use to identify the trajectories of brain development? So for this, I mean, it's pretty much stuff I just went over. Prefrontal cortex develops late, right? Sensory motor areas develop early, and then we use these longitudinal studies and the smooth curves that help you identify the trajectories of brain development. So I've posted all the things you need to know for the test online on Moodle. So there's a set of exam practice questions. There's a set of keys for exam practice questions. There's this whole review thing I'm going to put on Moodle, and basically everything except the test itself is on Moodle already. And so the format of the test is the same as before. I find that people are a lot less stressed if I just keep the same format, and by the third test they know what to expect, right? So there's 35 multiple choice, A to E. 35 points for short answers, which will be around 6 to 8 questions, sometimes with different parts. So just please do be guided by the little brackets at the end of the questions. Like if it says like 5 points, right, you should know that like I expect kind of 5 things. So they should be weighted. You should weight your time accordingly. Yeah, I hope that makes sense. Then there may or may not be a bonus question or 2 bonus questions. And then there's review session, which is on Sunday that Sean's going to run. Yeah, and then just like again, I mentioned this already, but if you need an extension for whatever reason, just arrange this with the class dean and your disabilities resources office. So Yeah, thank you for being so wonderful. A wonderful class. I've had a lot of fun. I know it's a difficult semester for me and for you guys for various reasons, but I mean, I really love this material. So, all right, so stay calm. Okay, great. So yeah, this is like I ended up way early, but I think that's okay.